So I guess everyone's here. Everyone's back. This light's really bright. Okay. So we're going to talk about the FoldTree, which is a really important data structure. It's pretty expressive, but it's really confusing. So without further ado, let's get started. We're going to kind of muddle up a little bit of the concept of it and a little bit of the detail of how it works. So hopefully that won't be too confusing, but we're going to do a lab afterwards, and that should help to clarify any of the problems that we have with the description that we're about to go through. So what is it? The FoldTree lives in core kinematics. It's used to specify geometric connectivity of the residues in a pose. And that might be different than the chemical connectivity. So that's the important thing here. And on a high level, it's a description of how to propagate conformational changes in a pose. So if I change a residue, we saw the atom tree. How do we build a new atom tree for everything? How do we get the new Cartesian coordinates out of that? So the major functional unit of a FoldTree is an edge. And there are two flavors that are really important that we're going to be discussing. The first is a peptide edge. And, oh, that's great. Let's get out of here. So they specify polymer connectivity in a shorthand form. They give a start and stop residues of a polymer chain, and no atoms are specified. It's just residues. Let's see if we can go back to a full screen representation. Wow. Okay, the second type of edge that's important is a jump. And that's how you can relate two local coordinate frames. So if you wanted to be able to say, starting at this residue, I want to tie it to this other piece of the protein in space. That's how you can specify this. And it's going to the edge contains the 3D transformation between those local coordinate frames and that are connecting edges of the graph. So the advantage of this is that we can describe connectivity and these relationships of these different parts of a protein without them needing to be chemically bound to each other. So here's a quick note on the edges. The order of the start and stop residues is important. It indicates the directionality of an edge. And if you add a whole mess of edges, there's this function called reorder. And you pass in a vertex where you want to start the FoldTree, and that'll attempt to make sure everything is in the right order. And if it can't, it returns false, and you can detect that and try and recover or not. All right, cut points. A cut point is a discontinuity in a FoldTree when you compare it to the polymer connectivity. The higher numbered end of a peptide edge is a cut point, always. And the highest number residue in the whole tree is always a cut point. But it is not counted if you ask for the number of cut points. So if you see that you have one cut point, that is excluding the last residue. And the number of cut points is always equal to the number of jump edges that you have because of these two facts. So what are some important methods of a FoldTree? Add edge.
I have a question about the cut points.
Yeah.
It seems like if you have a ring, there would be an edge in the chemical connectivity that's not in the FoldTree connectivity, but that's not a cut point.
An edge, how do you have a ring of protein?
A disulfide or something.
So disulfides are special.
But they're not cut points. That's the discontinuity. The cut point discontinuities are only when there is an edge in the FoldTree that's not.
So we build along the backbone, and the side chains come off. So the disulfides are a little different because of that.
Oh, okay. Well, what about like a cyclic peptide?
Those are special.
Okay.
People have to do a lot of work to make those work correctly.
Okay.
So those are not a general case for us. You can also talk about branching, and that's another problem that people have, and they put a lot of work into figuring out how to make that work. But that's just not part of the core basic functionality of the FoldTree.
What's the difference between a jump and a cut point? It's supposed to seem like something, there's a gap.
Oh, so a cut point is a node, and a jump is an edge.
Okay.
So they're just fundamentally different pieces of the graph.
So that's why it has a length you can specify. Right?
On the jump,
Yeah.
it has a rotation and a translation.
Right. That's right. I'm sorry. Rotation and translation.
Right. And the cut point is just a residue number.
Okay. Does it get added to the end? Is it like the end term, C term? Is like.
C term is a cut point, but basically what that means is that you don't take a change that happens there and pass it on to another residue.
You don't keep the change.
Like the change,
Okay.
because if you think about the last residue in the pose,
Right.
if it moves, nothing else is going to move. So that's
Cut point,
so
okay.
cut point. But we're going to go through some specific examples, and then the lab will really clear this up. You question?
No.
Okay. Okay. So to add a new edge, you just pass in the start, stop, and a label for the edge, and the label is going to be either this edge peptide or a jump, and jumps are specified with integers that are positive. So peptide edges are -1. Jumps are positive integers, so they start with 1 and just go up from there. There's this method called check FoldTree, and that returns true if you have a valid FoldTree, and we'll talk about what that means in a little bit, and false if you don't have one. Simple FoldTree, and you pass in a number of residues, and that just gives you a straight line. So that's nothing really exciting happens there. It's just the first, it's a single peptide edge starting with residue 1 and going to the last residue. So yeah, no jumps, no special features. And then you can make a new jump by specifying the start, stop, and the cut. And that does exactly what it looks like it would do. Okay, so we're going to talk about a couple of simple fold trees, and I'm using ASCII art because I love it. And so here's a really boring fold tree, right? It's the simple fold tree. So how many edges does it have? Okay, so I got n-1, and I've got 1.
You also got n.
And I got n. Well, if you add them together. Who thinks it's n-1? Okay, who thinks it's n? Who thinks it's 1? Okay. The ones have it. This is one edge, and it connects residue 1 to residue n.
Aren't there edges?
So basically, like the full tree is kind of like an abbreviated graph.
Okay.
It's
Not like a real graph.
it
It doesn't have nodes everywhere you think it has nodes. So it's yeah. So if
Got it.
so you can say that there's one peptide edge from 1 to n, but the real way I would think of it is there's an edge from 1 to 2, and 2 to 3, and 3 to 4,
Yeah.
all the way down to n, and then you'd have like the canonical definition of
Where I was going.
right, yeah. So in any tree, there's always n-1 edges.
Except the full tree.
So the fold tree is the fold tree is actually really disconnected from the pose. So that's the other thing. So if you notice in all those add edges and all that stuff, we didn't pass in a pose. It's not a member of a pose. Well, it's a member of a pose, but it's separate. You don't construct it from a pose per se. So because of that, there's really just these are the nodes, and there's one edge. There's only it connects this to this. Then when you map that back to a pose, it means something different. So it's different than the atom tree, which actually has this explicit connection to a pose. So that's kind of the confusing thing, but it allows you to talk about the way you would want to propagate certain changes in a pose at a very high level. Okay, so what might let's guess the number of edges in a fold tree like this, and also the direction of the edges.
Wait, what is that? What is that line there? Yeah.
It is a hat. That's why I was like, ooh, an ASCII hat.
Is that the cup one?
It's a cut point, yeah.
Yeah, I'll say Abraham Lincoln.
Okay, I should probably explain this a little more clearly. This vertical line here is a cut point, and this is a jump. So we're connecting residue 10 to 20.
Three edges.
Three edges then. What about one to end with three edges? Three edges.
I hear 1, 2, 3.
4 edges.
I hear 4. Do I have 5? And when I say edge, I mean peptide edge, not edge, peptide edge plus jump.
I think two.
Got 2? 2? Okay, so
Oh,
time for the big reveal.
oh, there might be.
We have an edge from 1 to 10. Another edge from 10 to 15. An edge going from 20 back to 15, actually goes to 16. And then from 20 all the way to n. Okay? So we have 4 edges from these residues. And we have a single jump. So we have 1 jump. So how many cut points do we have to have? 1, and it's 15. And this follows all those rules that we talked about earlier, because in 1 to 10, this is the higher number, but now it's the lower number here. So this isn't a cut point. And so this 16 is never, the 16 is a lower number, and this 15 is never visited in any other interaction. So this is the higher number in this, and it's not contained in another edge. So it's a cut point.
I thought you said the jump wasn't edge though.
Jump is an edge. So this, when I say edge, I mean peptide edge. That's the way that, yeah, not a general edge. I should have capitalized it, or italicized it, or quoted it or something.
Why is that line in one sentence an edge? Like in what sense?
So if I move residue 2, that means keep going this way.
Okay.
And then pop up here and move this, the rest. And then change this, and then change this. So it's telling you how to move the rest of the protein.
So basically, you have to have a node at 10 and a node at 20. And Brandon led but you also have a node at 16. So if you count the number of nodes, then the number of edges is that number minus 1.
Why wouldn't you have a node at 14?
Well, and so you go 10 to 15, and then you go 20 back to 16. But you don't have to stop at 14 along the way.
Right.
So it basically tells you how to close it.
Exactly.
So if I start at 1, I change everything to 10 then splits to 15 and 20, right?
Right, and also keeps going. 10 goes to 15, and then it goes directly to 20, and then 20 goes to n,
And
and
goes back to
16.
Why did 10 go to n and then all the way back?
Because it's acyclic. It's a DAG. So if 10 came back to here, then you would just kind of woo. And
It's free as opposed to a DAG, which is slightly more restricted.
sorry, it's better than a DAG. Yeah, so you can't visit anything twice.
Oh, we can't visit any twice. Oh, okay.
Right, because then if I move it and then I move it again, and then what does that mean about the first time?
Okay.
I think it becomes much more clear for people if you draw it really as a tree. If you don't hit the
Like a sycamore?
from 1 to 10, and then it splits, just as you said, it split left from 10 to 20, then it splits again from 20 to 50, and
Right. This sort of representation is what you'll see in publications where people talk about the connectivity. So I think it's important to bring it up. Yeah, I mean, I think that there's different ways to make sense of this, and this helps map that kind of a tree that isn't actually connected to the structure back to what's happening from 1 to n.
could you connect this to a change, like, so 1 to n is the linear order of the amino acids enclosed, and can you give an example of one where I want to impose like a cut point? Because if I change something that's like near 15, then I would expect it to go, I mean, it's obviously connected, right?
Sure. So the question is, why would you want this FoldTree?
Yeah.
Okay. Let's say there's a loop from 10 to 20, and I want to model that loop. If I move I know the rest of the protein, so 1 to 10 and 20 to n,
Right.
is right. Right. I don't want that to move. I just want to focus the movement of my loop to just the loop.
So you're constraining the residues between 1, 10, and 20 for your example.
Exactly. So in here, if you move this, it can only move this way, and then if you start at 19, it can only go this way.
All right. Yeah.
So then the rest of the protein that's not the loop stays fixed.
All right. So I have one more question.
Yeah.
So just building on what Tim said earlier, would you expect these lever arm effects to be worse at worse at residue like 9, at residue 14, at residue 16, and then at residue n, or something like that?
Worse at 9 than at 14?
Would you expect it generally to be worse at 14 than?
14 has a very small lever arm because it only goes to 15 and then it stops. It only moves one other residue.
Didn't you go from 10 to 14? No.
No, we're going from 10 to 15.
Okay.
So let's say at residue 11, this whole from 10 to 15 is just going to swing around. So those lever arm effects are kind of incorporated into this. Does that make sense? Like, we definitely have these lever arm effects, but part of the goal is to sort of restrict the part of the protein that we want to move around, and we tolerate those sorts of effects within that region.
Question.
Yeah.
So suppose back to your example that there's a loop between 10 and 20, and that you're trying to fold. Why do we even need the 1 to 10 and 20 to 4 at 20 to n edges? Like wouldn't we like never need that?
So the answer is that the FoldTree has to be able to hit every residue. So to be able to build an atom tree out of the FoldTree, you need to be able to start at some arbitrary point and root the tree there and then just go.
Never mind. I thought anyway. That wasn't even a valid question anyway, because you would still end up affecting the edges. Right. Okay. Never mind.
I mean, you need to.
Okay, yeah. Never mind.
That'll have to be there. It's fine. Any other questions? Okay, I've got more. So here's how you make this FoldTree. So we would start off by saying, I have a FoldTree named F. We're going to add edge 110, and we can say edge : peptide. And then we can add a jump from 10 to 20. We can call it jump num. In this case, it would be jump 1. And then we add another edge from 10 to 15, edge peptide, 20 to 16, 20 to n. And this edge peptide is a static constant integer defined as -1. So you could write -1 there, but we've kind of taken care of that so it's a little bit more readable so that you can say, oh, why is there this -1 floating around? Or you can say, oh, it's a peptide edge. Okay. So there's a couple of rules for FoldTrees. They must be connected. So you can't just have parts of a FoldTree that you can't get to or can't get out of. Every residue must be included. No cycles. It's a tree. And I guess think about what would happen if there was a cycle. You would update your coordinates and it would propagate it, and then it would go back to where you started, and it would just never really stop propagating that change. So that would be a big problem. Order matters because it's directed. And so, again, here's this reorder method that I pointed out before. And you pass in a start residue, which is the vertex of the tree. And then, so it roots it there and starts trying to make sense out of the edges that you have. And you can check for cycles, connectedness, making sure every residue is included with this check FoldTree. So we mentioned both of these earlier, but this is what they do and why they're important. So this is from a paper that you can't actually see the reference of, but you have the slides, with some commonly used FoldTrees. I have it written down what they all stand for, so I'm just going to pull that up. Okay. So the first one is called the protein folding FoldTree. It just goes from N to C.
What are the colors? Shaded versus non-shaded?
These are kind of like rigid edges versus things we want to kind of let be flexible.
Okay.
Quick question about A. So if that's a folding, like take a simple confirmation and just let it fold it,
Mhm.
it doesn't necessarily propagate linearly, right? I mean, I can change something in residue 20 and have it affect that residue, whatever, 75.
Right. So it's just saying that the
FoldTree should change as the confirmation changes, right?
if you that depends on exactly how your algorithm works and how confident you are that other pieces of the structure are correct and should be fixed in space. So, for instance, if you're doing a bunch of fragment insertion, you might want to let the C-terminal end of it just flop around and see what happens until when you get something with it that's compact. There are other situations where you definitely wouldn't want to do that. Right. Okay. So continuing, this would be like domain assembly. So you can see we have this fixed edge, and then we're going to put something in between and move it around, and then we're going to have this other fixed edge. So we have these two domains. We want to try to figure out how to move them around. So we're going to say this domain is fixed, that domain is fixed. Let's wiggle them around and not have these changes do anything other than move that entire chunk of protein.
The lines are the cut points, or no? No cut points.
So these lines are cut points.
Which one?
Like where that X is a cut point. So this is a loop modeling. This is the same one that I showed just previously in the ASCII art. So we have a jump here, cut point there, and we direct the change back. And this is the part we're modeling, the dashed line. Right. So this is the loop.
What is B?
B is domain assembly. So this might be a linker between two domains that we'd want to sample. Right, so then here's another loop in a different setup where we're not directing the other change back. We just have a giant lever for the loop. D is rigid body docking. So we pick the center of mass of one chain and jump to the center of mass of a different chain. Okay, so now if you move, what we're going to do is actually sample the translation and rotation from this and set it back and move the entire chain. And then this is docking with backbone flexibility. This is something more complicated where we start to have docking with a little bit of modeling on one side, and then this is docking and loop modeling at the same time. So you can see how you can start to just put these things together and kind of get additional complexity out of it. So I believe that's all of my slides. There's an activity. Was that shared yet?
Yeah.
Yep.
So let's get started with that, and hopefully all this stuff will make sense by the end of that.